Good morning, students. Welcome back to botany class. In a previous class, I started strategies for enhancement in food production. So there I started a plant breeding. So in a previous class, I explained main steps of plant breeding. After that, some hybrid varieties produced in India. Later, I started plant breeding for disease resistance. Okay. So in previous class, I explained you about the some of the microbes, particularly parasites, infects to the crop plants. When they infects to the crop plants, there is a great reduction in yield. Nearly 20 to 30% of crop is loss or sometimes total. Okay, so how we are going to produce disease resistant plants? To produce a disease resistance plants, we should know the causative agent and mode of transmission. If we don't know the mode of transmission and causative agent, we are unable to produce disease resistance plants. Okay, so here microbes such as bacteria, fungi, and viruses infects to the crop plants. So to kill bacteria, we are going to use chemicals bactericide, to kill fungi fungicide, and for viruses viruside. But these chemical substances are adversely affect on human and other organisms. Okay, so breeders they wanted to produce plants which are resistant to disease. Okay, here, what are the fungal diseases in plants? What are the bacterial diseases in plants? And what are the viral diseases in plants? In previous class, I explained you about the viral diseases, fungal diseases in plant. So, what are the fungal diseases in plants? Fungal diseases in plants are brown rust of wheat, brown rust of wheat. The second one is the red rot of sugarcane, sugarcane. Then the third one is the late blight of potato. Okay, so what are the fungal diseases in plants? Fungal diseases in plants are brown rust of wheat, red rot of sugarcane, and the third one is the late blight of potato. So many more fungal diseases in plants, but as per your NCERT text, they are mentioned three fungal diseases. So these three you have to remember. Is it clear? In even in theory, they are going to ask that name two fungal diseases in plants, name two bacterial diseases in plant, name two viral diseases in plant. Very very important for 1 mark. Okay. So moving to the bacterial diseases in plants. So what are the bacterial? So many are there, but only one they are given here. That is the black rust of crucifer. Black rust of crucifer. Okay. Uh, this is caused by the one of the bacteria. Is it clear? It is caused by this disease is caused by the bacteria. Is it clear? Which disease? Black rust of crucifer. Is it clear? Actually, it is caused by one of the bacteria, that is the Brassica compastris. Is it clear? It is caused by the bacteria, that is the Brassica compastris. And moving to the third one is the viral diseases in plants. So what are those? The first one is the tobacco mosaic. tobacco mosaic, and the second one is the turnip mosaic. Turnip mosaic. Turnip mosaic. So, what are the viral diseases in a plant? So, the viral diseases in plants are tobacco mosaic, and the second one is the turnip mosaic. So, tobacco mosaic is caused by the virus that is called as a short form TMV. Tobacco mosaic virus. Is it clear? And turnip mosaic virus. So these are the some of the fungal virus as well as the bacterial diseases in plants. So how we have to produce disease resistance plants, so that we are going to study now. Now we are going to study methods. So the next heading is here the methods of plant breeding for disease resistance. So breeding is carried out by two methods. One is the conventional conventional and another one is mutation breeding. Mutation breeding. So methods of a plant breeding for disease resistance. So breeding is carried out by the two methods. One is the conventional method and another one is the mutation breeding. So convention breeding, so this is through hybridization as well as the selection, and here it is similar to that of the what steps which are involved in this conventional method is the steps which are involved in the main steps of plant breeding. So here we are going to study about the what are the sequential steps of a conventional method. Sequence sequential steps of conventional breeding. The first one is here screening, germplasm for resistant source. The second step is here hybridization. Hybridization of selected parent. Then third one is the selection and testing of selection and evaluation of Okay, and the last one is the so there are four steps or the sequential steps of plant breeding. The first one is the screening germplasm for resistant source. The second one is the hybridization of selected parent. And third one is the selection and evaluation of hybrid. And fourth one is testing and release of new varieties. Okay, so here these are the four steps of conventional breeding method. So you know very well already was studied in main steps of a plant breeding. What are the main steps of plant breeding? Collection of variability, selection and evaluation of parent. Then third one is the cross hybridization among selected parent. Okay, and then the evaluation and testing of superior recombinants, and the last one the testing, release and commercialization of new varieties, new varieties. Is it clear? So you are studied the five main steps of a plant breeding. So same methods here also they are going to follow screening germplasm for resistant sources. Here, what they are going to do here, in main steps, you have studied the first one, collection of variability. The genetic variability is available in wild varieties, species, and its relatives. And these variations are heritable. The naturally occurring genes in these plants control their character. Is it clear? So, here in this first step, we are going to collect some plants. Those, for example, I am collecting plants. All four plants they are genetically differ from each other. One plant is rich in a disease resistance, another plant is rich in a protein, another plant is resistant to the insect, and another one is rich in what the it gives high yield. So all four plants differ from each other. Is it clear? The entire collection of plant or seeds having all diverse allele for all genes in a given crop is called as germplasm collection. So in this, we are going to study about the screening germplasm for resistant sources. We have to screen means screening germplasm for resistant source. After that, what we have to do, we have to cross those plants. Is it clear? Suppose we have to select two plants which have desirable characters. Is it clear? Those plant we have to cross. Hybridization of selected parent. Is it clear? In a first step, we are selecting the plants. Is it clear? Which have a desirable character, and those plants we are crossing. Is it clear? So they said that hybridization of, of selected parents. In the previous classes said that what is meant by hybridization? A cross between two plants differing each other genotypically in one or more characters to produce fertile offspring is called as hybridization. Is it clear? So here the what plants we are selecting, those plants we have to what cross. Is it clear? So that is the second step, hybridization of selected parent, and the third one, selection and evaluation of hybrid. So when the progenies are formed, is it clear? For example, this is a one plant, this is another plant. So these plant we have to what cross, and when we are crossing, we will get so many plants. Is it clear? Maybe in a previous class I told that hundred to thousands of progenies are formed. In that progenies, only one plant have a desirable character. So that plant we have to select. Is it clear? Selection and evaluation of hybrid. Is it clear? So that plant we have to select, and that plant we are going to what? Evaluating. After evaluating that plant, we are going to what? testing. Is it clear? So testing in a research field, then testing in a farmer's field, in a three different seasons, in a different parts of the countries, and then those seeds are comparing with the best local available seeds. If it is a good, then that new variety of seeds are releasing into the where market. Is it clear? So only they said that selection and evaluation of hybrid. Then fourth one is the testing and release of new varieties. Means if it is a good, then we are releasing into the where market. So these are the what some of the sequential steps of a conventional method of breeding. Okay. Next one is here. What are the uh, uh, crops which are produced and so scientists or breeders they produced some of the plants which are resistance to fungal viral as well as the bacterial diseases. So we'll study about that one now. So what are the varieties? Okay, the first one, first column, crop. Second one is a variety. Third one is resistance to the disease. Okay, first one is a crop. Which crop here? First crop name is wheat. Wheat. Okay, the first crop is a wheat. Variety is Himgiri. Himgiri. And this variety is resistant to. So one of the crop plant that is the wheat, it is resistant to leaf and stripe rust, hill bunt. The name of the variety is Himagiri. Is it clear? Himagiri is a variety of wheat resistant to leaf and stripe rust, hill bunt. So this it is a fungal disease or the bacterial disease or the viral disease. So this is a fungal. Is it clear? So wheat it is a resistant to the which disease? Resistant to the leaf and stripe rust, hill bunt. Okay. Moving to the second crop. Second crop is here brassica. Brassica. It is also called as currant rye. It is also called as currant rye. Okay. So variety of brassica is Pusa sornim. Pusa sornim. Okay. So this variety is resistant to which disease? It is resistant to white rust. it is resistant to white rust. Is it clear? Second crop plant, that is the brassica. Brassica is also known as the currant rye, and this crop is resistant to the white rust, and the variety of this one is Pusa swarni. Okay. The third crop is the third one is the cauliflower. Cauliflower. Okay. So the variety of the cauliflower is Pusa shubra or Pusa snowball K1. Pusa snowball K1. So here the another or the third crop name, that is the cauliflower, and it is variety of the cauliflower is Pusa shubra or Pusa snowball K1, and it is resistant to black rot, curl blight, black rot. The third one is the cauliflower. It is resistant to black rot, curl blight, black rot, and the variety is Pusa shubra or Pusa snowball K1. Okay, so it is one of the bacterial disease. It is one of the bacterial disease. So cauliflower is resistance to which disease? It is resistance to black rot, curl blight, black rot. Okay, is it clear? Moving to the next one is the fourth one. The fourth crop plant is cowpea. Cowpea. So the variety of the cowpea is Pusa komal. Pusa komal, and it is this variety is resistant to bacterial blight. Bacterial blight. Is it clear? So next crop plant, that is the cowpea, and it is resistance to the Pusa komal, resistance to the bacterial blight, and variety is Pusa komal. Okay. Moving to the last crop name, fifth one, that is the chili. Chili. So the variety of chili is Pusa sadabahar, sadabahar. It is a variety of chili, and it is resistance to chili mosaic, chili mosaic, tobacco mosaic, as well as leaf curl. Leaf curl. So the next one is the chili. Chili, it is resistance to the chili mosaic or the tobacco mosaic and even leaf curl. So the variety is Pusa Sadabahar. Okay, so these are the crop plants variety and resistance to the bacteria, fungal as well as viral diseases. So Pusa Sadabahar, you know very well. Cowpea variety is Pusa comal. Resistance to disease is a bacterial blight. It is one of the bacterial disease only. And chili, Pusa Sadabahar, it resistance to disease is chili mosaic, tobacco mosaic, leaf curl. It is a what? Viral disease. Is it clear? So these are the varieties and resistance to the disease. Here you can be observed that here actually five examples they are given as per your NCERT text. Variety actually it is starts from what? Pusa, Komal, Pusa Sadabahar, Pusa Shubra, Pusa Swarnim. So why they are given? Indian Agricultural Research Institute, short form we are going to call as the IARI, Indian Agricultural Research Research Institute, where it is located in New Delhi. IARI has given the name Pusa. Is it clear? It is a Pusa is a variety given by IARI, that is the Indian Agricultural Research Institute. Okay, so today we are studied about the some of the varieties which are resistant to the bacterial, fungal as well as the viral diseases. Okay, so you have to remember very important for the NEET as well as for the theory. Name the variety of wheat resistance to leaf and stripe rust hillbunt. Which variety of wheat is resistant to this? Variety is Himagiri. Is it clear? Same thing is here. Second example, you are studied about the brassica. Variety of the brassica is the Pusa swarnim, resistant to the white rust. Then you are studied about the cauliflower, that is the Pusa shubra or Pusa snowball K1, resistant to black rot, curl blight, black rot. Then moving to the fourth example, they are given here. Cowpea varieties Pusa comal and resistance to diseases, bacterial blight. Then moving to the fifth one, chili, that is the what? Pusa sadabahar, or it is resistant to diseases, chili mosaic, or the tobacco mosaic, or leaf curl. It is one of the viral diseases. Disease. Okay, is it clear? So this is about the brief introduction about the conventional method. Moving to the second method, that is the mutation breeding. Moving to the second method for producing disease resistance, that is the mutation breeding. So what is meant by mutation breeding? Mutation breeding means what? Before studying the mutation breeding, first one, what is meant by mutation? What is a mutation? Mutation means sudden change. Sudden change in the structure of DNA. Sudden change in the structure of DNA is called mutation. Is it clear? Mutation breeding technique was first introduced in India by the scientist M.S. Swaminathan. M.S. Swaminathan. Scientist Dr. M.S. Swaminathan, he first introduced this technique in India. Is it clear? Mutation breeding technique was first introduced in India by scientist Dr. M.S. Swaminathan. Okay, mutation means sudden change in the structure of DNA, that is called as the mutation. Okay, here we are going to study seeds. They in this method, seeds are exposing to mutagens. Seeds are exposing to mutagen. So here now question is here, what is meant by mutagen? What is meant by mutagen? Is it clear? In mutation breeding, seeds are exposing to mutagen. But question is here, what is meant by mutagen? Mutagen means agent cause mutation is called as the mutagen. Is it clear? Agent cause mutation is called mutagens. Is it clear? So here mutagens may be physical or chemical. Mutagens may be physical mutagen or maybe the chemical mutagen. So what are the physical mutagen here? So the physical mutagens are gamma radiations, as well as UV radiations. These are the examples for the physical mutagens. Is it clear? It means that seeds are exposing to gamma radiations or UV radiation. When they are exposing to such radiations, so what will happen there? Sudden change in the structure of DNA. Is it clear? That results in the formation of a variety of seeds which are resistant to disease. Is it clear? So here physical mutagens are gamma radiation as well as the UV radiations, and chemical mutagens are nitrous acid, chemical mutagens are examples are the nitrous acid or ethyl methane sulfonate. Short form we are going to call as EMS, ethyl methane sulfonate. So these are the chemical mutagen, means seeds are exposing to the what? Chemical substances. Due to that, sudden change. Is it clear? So here the mutagen may be the physical mutagen or maybe the chemical mutagen. Okay. Moving to the next one is here. Okay, moving to the next one is here the what is now question is here, what is meant by mutation breeding? So we are studied about the mutation, what is meant by mutagen and what are the mutagens we are studied. Now we are going to study about the what is meant by mutation breeding. So mutation breeding means improvement of crops by changing the genotype of a plant by induced mutation is called as mutation breeding. So what is meant by the mutation breeding? Improvement of crop plants by changing the genotype of plant by induced mutation is called as mutation breeding. Is it clear? So your improvement of crop plant by changing what? Genotype. Genotype means here the genetic constitution of plant. You know very well which is the genetic material which is present in most of the organisms, that is the DNA. So DNA is the genetic material present in most of the organisms. Is it clear? So here what they are going to do here, they want improvement of a crop plant by changing what? The genetic constitution of plant by induced mutation. It means that exposing the seed to physical mutagens or maybe the what? Chemical mutagen. They are altering or they are changing the genotype of the plant. So that is called as mutation breeding. Okay. So like one of the two examples they are given here. What are those examples? The first one example they are given here. Here some example they are given. The first one is here. In moong resistance to yellow mosaic. It's a very important example for the mutation breeding. Moong bean is resistant to yellow mosaic and powdery mildew. This is one of the example for the mutation breeding. Understood here? Remember it is very important for 1 mark. Mung bean is resistance to yellow mosaic and powdery mildew. Okay. Second one is here some of the wild varieties and their relatives have uh, desirable genes. They have what? Desirable genes, but they give less yield. Is it clear? They gives what? Less yields. Some of the wild varieties and relatives, they have a desirable gene, but gives less yield. Is it clear? So such genes, they are transferring into the other plants. Okay. For example, one of the example they are given here, the bindi. bhendi or bhindi. Okay, you know very well scientific name, that is the Abelmuscus esculenta. Abelmuscus esculenta. Okay, Abelmuscus esculenta. So it is one of the bhendi. Okay, so in this they are introduced a wild variety gene, and there's a formation of a new variety of the bhendi, that is the so the new variety of the bhendi, that is the Parvane Kranti. Is it clear? So this is the one of the new variety of the bhendi which are formed by this one. Which one is the gene transferred into this bhendi, Evalmuscus esculenta? It results in the formation of a one new variety of bhendi, that is the Parvane Kranti, and it is resistant to the what? Yellow mosaic. It is resistance to yellow mosaic. Yellow mosaic. Is it clear? So these are the examples for the mutation breeding. In the mung bean is resistance to yellow mosaic and powdery mildew, and name the variety of bhendi which is resistant to the yellow mosaic, that is the Parvane Kranti. Is it clear? So this is about the brief introduction about the methods of breeding for disease resistance. Is it clear? After this, we will go to the next one is the after the conventional breeding method as well as the uh, second one is the mutation breeding. We'll go to the plant breeding for developing. Okay, till now we have studied about the high yield production of high yield as well as production of disease resistance plant, and the third importance now we are going to study here the resistance to insects. Is it clear? So the third importance is plant breeding for developing insect resistance plants. Is it clear? It is another one due to the infection of insects. There is a large destruction of yield. Like a bacteria, fungi, as well as the viruses, insects also infects crop plants, and there is a large destruction in yield. Is it clear? So how they are going to produce insect resistant plant? It is due to the morphological, physiological, as well as the biochemical characteristics. It is due to the morphological, biochemical, as well as the physiological characteristics. Is it clear? Like a bacteria, fungi, and viruses, insects also infects plants, and there is a large destruction in yield. So that is a prevented morphological, biochemical, as well as the physiological characteristics. Okay, so here the first one we are going to study here So here the first one is here plant breeding for developing insect resistance plant. Hairy leaves is associated in many plants to produce insect resistance plants. Okay, for example, they are given resistance to acids in cotton, resistance to acids in cotton, resistance to acids in cotton, then cereal leaf beetle in wheat, cereal leaf beetle in wheat, solid stem, non-preference by stems of fly, and fourth one is here the smooth-leaved and nectarless cotton varieties do not attract bollworms. Then last one is here the high aspartic acid, low nitrogen, low sugar content, maize, high aspartic acid, low nitrogen, and low sugar in maize, resistance to stem borer. So hairy leaves is associated with several plants which is resistance to the insect. So some of the examples they are given, the resistance to acids in cotton, and second one is the cereal leaf beetle in wheat. Resistant to what? Cereal leaf beetle in which plant? Cereal leaf beetle in wheat plant. Okay. Third one is in wheat. Solid stem. Is it clear? Solid stem, non-preference by stem soft fly. They're not going to prefer what? Stem soft fly. Is it clear? So these are the stem sawfly are the insects which are going to infect to the water wheat. But due to this character, they produce the plants which are resistant to the insect. For example, wheat in a wheat solid stem non-preference by stem sawfly. Second one is here the smooth-leaved and nectarless cotton varieties. Nectar meaning is here the sugar. Nectar means here the sugar. Okay, sweet material that is called as the nectar. Smooth-leaved and nectarless cotton varieties do not attract to the bollworm. Is it clear? Very, very important. These all very, very important for NEET. You have to remember it here. Smooth-leaved and nectarless cotton varieties do not attract to bollworms. Is it clear? And fourth one is here the high aspartic acid. Aspartic acid, low nitrogen, low sugar, in maize, resistance to stem borer or maize stem borer. Is it clear? You have to remember it. These all examples very, very important. As per your NCERT text, these 5 examples they are given, you should be remembering. Okay, is it clear? So here, after this, they are given some of the plants which are resistant to the insects. So, what are the examples? So, here some of the crop plants which are resistant to the insects. The first crop name is Brassica. Brassica. Okay, it is a crop name, and it is the name of the variety is, it is also called as the rapeseed. Okay, and variety of the Brassica is Pusa gaurav. So Pusa gaurav, it is a variety of rapeseed. It's a Pusa gaurav is a variety of rapeseed, and it is resistant to the aphids. Resistance to the what? Aphids. Moving to the second example, flat bean. Variety is Pusa sem2, Pusa sem3, Pusa sem2, Pusa sem3. Resistance to jacids, aphids, and fruit borer. And fruit borer. Fruit borer. Is it clear? Flat bean. So variety is Pusa sem2, Pusa sem3. Resistance to which one is jacids, aphids, and fruit borer. Is it clear? So the in the next class I will be explain you about the another one example, that is the okra or bendi. So the variety of the bendi is the Pusa savani. So that I will be explain you in next class. Okay. Thank you.